crossovers generate non-random recombinants under Darwinian selection. The title of this section is the title of one of my earlier published contributions to evolutionary theory. In addition to helping to clarify the nuances of the roles played by recombination and point mutations in DNA sequence evolution, the paper exemplifies the mutually beneficial ways in which basic research, applied research, and theoretical research interact, leading to new experimental designs and surprising results. I'll begin with an excerpt from the paper's abstract. Summary: genetic diversification is not the only consequence of recombination. When recombination occurs in a gene pool under selective pressure, the result is non-random linkage of selected sequence elements with high potential fitness. Computer simulations based on the genetic algorithm show that populations of DNA sequences evolve more rapidly via homologous recombination than by point mutations alone, especially under mild selective pressure. The paper illustrates the way that a technical innovation, the GA, inspired by neo-Darwinian variation and selection, led to a computer simulation designed to learn more about the natural system that inspired the technical innovation in the first place. Briefly, I systematically compared the effects of simulated point mutations, single nucleotide changes, to recombination, reciprocal or unidirectional exchanges between sequences, as well as simulations in which both point mutations and recombination were allowed to take place. Various degrees of selection were simulated by selecting sequences for the presence of codons for an arbitrary amino acid, lysine, and then randomly replicating the selected sequence fractions to restore the original populations in each round. Interested listeners are urged to download and review the original paper. Briefly, these simple simulations, carried out on an antiquated desktop computer with a tiny fraction of the computing power of a smartphone, demonstrated why and how, contrary to the precepts of the modern synthesis, recombination can represent a potent creative force in natural selection. In fact, the combined effects of recombination and point mutation, along with other sources of DNA sequence variation, play a variety of synergistic roles in actual DNA sequence evolution that has been thoroughly documented in numerous empirical studies. Chapter 16.